In this video, we're going to be doing a brief introduction to two broad categories of your circulatory system's white blood cells. Technically, I misspoke there because unlike red blood cells that have the primary function of transporting oxygen and carbon dioxide through your body, white blood cells are more accurately grouped with the immune system rather than the circulatory system, even though they do use it for the purpose of getting around the body, because the primary function of white blood cells is defensive rather than transportation, in that the function of your white blood cells is to hunt down and destroy any pathogens throughout the body that can make you sick. And in this video, we're going to be looking at two broad categories of how white blood cells actually defend your body against pathogens, starting with white blood cells that use a technique called phagocytosis in order to destroy bacteria and viruses. Phagocytosis is just a complicated word that literally translates as cell eating. So cyto is the prefix that refers to cells, and phago, sometimes pronounced phago, is the process of eating. So literally, these examples of white blood cells will move through your circulatory system, and when they encounter something that is hostile, they will literally engulf whatever that is with their cell membrane and internalize it using their lysosomes to destroy the pathogen once they have internalized it. So the category that we often use to describe this type of white blood cell are what we call phagocytes. Now, phagocyte is not actually a specific classification. White blood cells can be even more specifically classified, but it's more a description of their behavior and how they actually destroy the pathogens that they encounter. Now, because phagocytes need to get close to pathogens to actually destroy them, because they're literally eating them, phagocytes by necessity are highly mobile. Not only can they use your larger blood vessels like arteries and veins to freely move around the body, but they can actually extend and contract their cell membrane to basically crawl along the walls of your organs and blood vessels, as well as use a technique called extravasation in order to actually move or in order to actually transport themselves through different layers of cells. So in the event that a bacterium gets out of the blood and into some type of tissue, the phagocytes can actually use extravasation in order to basically squeeze through the cells that make up the wall of a blood vessel in order to be able to follow the pathogen to where it is going. The problem is that phagocytes need a way to distinguish what a pathogen is and what makes a pathogen different in structure to the normal cells within their body, which brings us to the second category of white blood cells that we're going to be talking about, which are what we call B cells. So B cells were introduced briefly when we talked about blood typing, because B cells are specialized white blood cells who exist as mobile factories that produce specialized proteins that we call antibodies. And we'll talk about what antibodies actually do after getting a brief look at what B cells are. So B cells are so specialized that they actually begin inactive. Most of the B cells in your body are basically just sitting there waiting to be activated, because the amount of energy that B cells require in order to produce antibodies is quite substantial, and therefore keeping them on all the time is not an efficient use of the body's energy resources. However, B cells become activated when the proteins, specifically receptor proteins on the surface of a B cell, recognize when a specific antigen is present. So an antigen is a specific compound or a specific part of a pathogen that has the ability to activate a B cell and cause it to start producing antibodies. And when B cells become activated, they become what we call plasma cells. Now, we call them plasma cells because remember that plasma is the liquid part of your blood, and we call them plasma cells because B cells begin massively producing proteins that we call antibodies that then make up a significant percentage of the plasma in your blood. And because your blood is constantly moving, this circulates the antibodies around the body, and eventually we'll see what these antibodies actually do. Now, the odds of a specific pathogen encountering this specific B cell that recognizes a specific antigen on that pathogen is relatively low. So a lot of the time, phagocytes actually serve a different type of function. So sometimes simpler quote-unquote white blood cells, including phagocytes, actually float around the body and look for specific pathogens, and if they break up a piece of that pathogen, they can actually travel to B cells where they basically show the B cell, hey, I found this piece, this compound on a pathogen. I think this is the antigen that you can recognize, and this is the process that we call antigen presentation, which actually allows quote-unquote simpler white blood cells to activate a greater number of B cells because they found a specific antigen when they were hunting down pathogens in the body, and this activates a greater number of B cells in order to start producing antibodies, which brings us to what do antibodies actually do. So antibodies are instantly recognizable by their classic Y shape like this, and the ends of the forks of the Y are the actual active site of an antibody, and this active site, it has a specific structure that allows it to recognize the specific antigen that activated the B cell that produced these antibodies. And basically, these antibodies with their specific active sites will specifically float through the blood and bind to the antigen on whatever pathogen contains that. And there are a few consequences that result. So we can see here that multiple pathogens, each of which that contain the antigen, the target that the antibody binds to, are linked via antibodies like this. So basically we see here that one antibody has attached to the antigen on this pathogen here, and a second antibody has linked to this pathogen here, which causes the pathogens to clump together, which makes it a lot harder for these pathogens to move. Now, this process here is what we call pathogen agglutination. So we can say that these pathogens have agglutinated, which makes it much easier for phagocytes to actually grab onto these pathogens because, again, not only can they not move, but now there are also multiple pathogens that are trapped together, which makes it easier for a phagocyte to grab onto multiple pathogens, eat them, and destroy them all. Now, the antibody itself also basically serves as a giant recognition molecule that makes it easier for phagocytosis to happen. That being basically each of the antibodies, as soon as I write phagocytosis here, each of the antibodies basically acts as a giant signaling molecule that makes it easier for your white blood cells to destroy that pathogen via phagocytosis. So this process of antibodies flagging a pathogen and basically alerting all of the white blood cells that can destroy it more easily, this process is what we call opsonization, which again makes it easier for your phagocytes to target a specific pathogen. Firstly, because the pathogen has been immobilized, because it's a lot harder to move around if it's covered in antibodies. And secondly, because your white blood cells specifically recognize the other end of antibodies and can grab onto the antibodies and the pathogen more easily, internalize it, and then destroy it once it is inside. In the next video in this series, we're going to take a more detailed look at a specific role of antibodies and how it ties to blood type identification and what exactly blood type identification is in terms of the difference in structure between red blood cells of different blood types.